Okay, this is lecture, part 2 of our chapter 20, material antimicrobial medications. We're on slide 12. We're looking at drugs that inhibit bacterial cell wall synthesis. So the first few slides here, slides 13, 14, 15, 16 and 17 and 18, we've looked at those before. These are just understanding what peptidoglycan is, looking at the difference between the structure of a gram-positive cell and a gram-negative cell. So just a very brief, we have our cytoplasmic membrane. That's the membrane outside of the cytoplasm. And if we are a gram-positive bacteria, we have lots of layers of peptidoglycan, and that's it. So a thick peptidoglycan cell wall defines a gram-positive. And gram-negative, they start with their cytoplasmic membrane. They have a couple of layers, a thin layer of peptidoglycan, and then on the outside they have that outer membrane. Let's look at slide 18, because this will be a little bit important later on. The idea that gram-negative bacteria are typically more resistant to drugs than their gram-positive cousins. Because in this first section, clearly our target is the peptidoglycan. On our gram-positive bacteria, the peptidoglycan is right there to be attacked. Our gram-negative bacteria, the peptidoglycan is shielded by this outer membrane. So if we are attacking peptidoglycan on gram-positives, it's very easy. If we're attacking peptidoglycan on gram-negatives, we have to get our drug across the outer membrane, and then we can start attacking the peptidoglycan. And that can be a problem, because the bacteria is in charge of what goes, or typically in charge of what goes backwards and forwards across the outer membrane. They can have these protein channels, and the presence of, say, antibiotics in the environment can affect those protein channels. So you should know what peptidoglycan is. You should be able to identify a gram-positive and gram-negative. This is slide 19. You should know what LPS is, lipopolysaccharide, comprised of a couple of different parts of stuff, lipid A and O antigen. For example, E. coli O157H7, the O157 part. That's the O that talks about the O antigen. Lipopolysaccharide, we mentioned that before. If you recall, in our look at the immune system, lipopolysaccharide is one of those things we called a T-independent antigen, in that it could attach to B cells, and it could trigger those B cells to start producing antibodies without the need for helper T cells coming in and activating those B cells. So in a lot of ways, an infection with gram-negative bacteria can be more serious. If they get into the blood, they trigger the immune system very quickly. Now, also there are a large number of very nasty gram-negatives too. But hopefully all that stuff about peptidoglycan and cell walls and cell membranes should be just a review of material we've looked at before, not only in lecture but also in lab. So let's move on to slide 20. Let's look at the drugs themselves that affect the bacterial cell walls. We have those drugs that inhibit cell wall synthesis. In particular, we can look at what are called beta-lactam antibiotics, such as penicillin, cephalosporin, and carbapenems. And then we have other drugs that affect cell walls, such as vancomycin and bacitracin. So, let's start with our beta-lactam antibiotics. And that's what that funky looking B symbol stands for. It stands for beta. Beta-lactam antibiotics. You'll see, if you look in the book and look on the slide there, on slide 21, we have a simple chemical structure for our penicillins and our cephalosporins. And you'll see right in the center there is a square component to our drugs. There's some stuff over here, there's a square thing in the middle, there's some other stuff in different parts of the drug. And we see that this square component here is present in our penicillins, it's present in our cephalosporins. This square thing here is called the beta-lactam ring. And they've represented it as a ring in the book there. And this is what makes our beta-lactam antibiotics beta-lactam antibiotics, because they all have this beta-lactam ring. So what do? They inhibit the formation of peptidoglycan. In particular, they block the formation of these things called peptide bridges. So we have our peptidoglycan, our NAM-NAG chains, our sugar chains, and then in order for our layers of peptidoglycan to actually hold together and be stable, there are these bridging structures that link the chains together. These bridging structures make the whole thing strong and rigid. And these bridging structures are built by enzymes. And if we interfere with the ability of the enzymes to build these bridges, then they don't get built, and the bacteria starts building very weak cell walls. There's pressure inside a bacteria. It's been described as about the pressure inside a bike tire. So what happens if you weaken the rubber of a bike tire? It's going to pop, and all of the stuff inside is going to squirt out. And that's exactly what's going to happen with our bacteria. A bacteria with a weak wall is going to burst, and all of the stuff that was inside the cell is going to spew outside of the cell. So that's always going to be destructive to the cell. So look at slide 22. What we have are enzymes that build these bridges. And these enzymes have an active site. And that active site works to bind its substrate, and then the enzyme has a chemical reaction, and through that chemical reaction the bridges are built. So what we're doing with our beta-lactam drugs is we are jamming up the active sites. We are doing what is called competitive inhibition of those enzymes. We looked at the idea of competitive inhibition before. So here's our enzyme. Here's the active site. This is the drug that the enzyme wants to work with, and it can work with that, and it can build a wall. If we put in a drug that is similar enough in shape, it jams up the active site. It prevents the enzyme working with its substrate, and then the enzyme can't build those walls. So this is where that idea of shapes is very important. Our enzyme is a protein. Our protein is all folded up into that three-dimensional structure, and it has an active site. That active site has a particular shape because the shape of its substrate is going to fit into that active site. We can gum up the active site with our kind of fake, with our drug, then the enzyme's activity has been destroyed. So our drug has to be similar enough to the enzyme's real substrate, and that's where the beta-lactam ring comes into effect. It is that beta-lactam ring that is sticking into the active site. It's the beta-lactam ring that allows the drug, the beta-lactam drug, to clog up those active sites. So those enzymes that want to build cell walls, they want to use their substrate and build walls. These are known as penicillin binding proteins, because when scientists were first discovering all of the chemistry of bacteria, they saw that these enzymes were binding penicillin. The downside to calling these penicillin binding proteins is it sounds like that's what they're designed for. That's not the case. They are not designed to bind penicillin. So currently PBP stands for penicillin binding protein. That's what it stands for. I'm probably going to spell penicillin wrong many times. But we can perhaps think of it as our peptidoglycan building proteins. That's what they actually do. They build peptidoglycan. So just remember what PBB actually stands for, and then maybe you can remember a more helpful name for it. Okay, so penicillin binding proteins build walls. The idea is then you interfere with the building and the cell builds bad walls. So on number 23, there, slide 23, penicillin is a very obvious example of a beta-lactam antibiotic. And it does do exactly that. It interferes with the ability of bacteria to build cell walls. And that's the important part. It interferes with the building of the cell walls. If the cell wall is already built, it can't knock it down. It can't damage an already built cell wall. So this is where you can imagine somebody is out there building a brick wall, and they're putting the bricks, and they're putting the mortar on the bricks, and they're putting the bricks in order. If you were to go over there and just watch them build the wall and not interfere, that wall is built and it's good. You can't do anything against a wall that's already built. But if they go on to the next wall and they're trying to build that second wall, and you keep on, say, breaking their little shovel, that's going to interfere with the ability of that person to build that wall. And you keep on messing with them, keep on breaking their tools, keep on stealing their bricks, keep on kicking over their bucket, they're going to build a weak wall. And that's what we're doing. We're interfering with the process of building that wall. And that's why these drugs are particularly effective, really only effective against growing cells. We have our phases of bacterial growth: the lag phase, the log phase, stationary phase. It's in the log phase where most, or indeed most, the building part takes place. And this is where they're building things fast. This is where they're very vulnerable, because if you start to interfere with those processes enough, they're already building walls very fast. If we interfere with that building process, they're going to build lots and lots and lots of bad walls, and that's going to lead to a lot of burst cells. This is why some bacteria are not affected by penicillin binding proteins. In particular, a bacteria such as Mycobacterium. Mycobacterium tuberculosis is a very slow growing bacteria. So it typically is not affected by penicillins and beta-lactam drugs because of that slow growth. Look at number 24, slide 24. We have our gram-positive and our gram-negative bacterial cell walls. And then hopefully you can see if we apply our beta-lactam drugs to a gram-positive and a gram-negative, its ability to affect a gram-positive is much more pronounced, because the cell of a gram-positive is building its walls with no defense. Now imagine our worker there building their walls, and we can go up to them, we can interfere with that process, but imagine then they are building those walls behind a fence. We could get to the fence, and we couldn't then get past that fence to go and interfere with that workman. So the gram-negative outer membrane is very important in the ability of beta-lactam antibiotics to affect cells. Not just beta-lactam drugs, the outer membrane of gram-negatives has an effect on many kinds of different drugs. Also, the penicillin binding proteins in gram-positives and gram-negatives are different. Another thing that is important to understand when we're trying to target a cell with a drug is its biochemistry. Is it aerobic? Does it use oxygen? Is it anaerobic? Those two different kinds of bacteria have different requirements. They have different abilities to move things in and out of their cells. So, on to slide 25. We are attacking the cell's penicillin binding proteins with drugs. And the important part of that drug is that square, the beta-lactam ring. So this is clearly bad for the bacteria, and the bacteria know it. Now, bacteria have been fighting each other, and they've been fighting with other organisms in the environment for a very, very long time. So they have, through evolution, they have these abilities to fight back against drugs. One important way that a bacteria can fight back against a drug is by making an enzyme that snips that drug apart. And that's our next topic of beta-lactamases. So ends in A's. Therefore we know it's an enzyme. Therefore we know it's a protein. And beta-lactamases are enzymes that are made by the bacteria. The beta-lactamase recognizes the beta-lactam ring and it cuts it. So our beta-lactam drug is snipped, and that snipped version of the drug can no longer stick into the active site of our penicillin binding protein. So our penicillin binding protein keeps on accessing its substrate, keeps on building walls, and our penicillin binding protein, after it's been snipped, can no longer interact with the active site of that penicillin binding protein. Typically, gram-negatives produce a more extensive array of beta-lactamases than gram-positives. Different kinds of beta-lactamases can affect different kinds of drugs. These things called extended spectrum beta-lactamases, or ESBLs, can inactivate a wide range of beta-lactam drugs. A beta-lactamase that is specific to deactivating penicillin is called a penicillinase. On slide 26, there are many examples of different kinds of penicillin on page 507. One example is methicillin. Methicillin is a type of penicillin, and methicillin was developed to counteract penicillinases. So penicillinase resistant, in that penicillin A's, which snips up other kinds of penicillin, won't snip up methicillin. It was common to treat S. aureus infections. So, since the use of methicillin to treat S. aureus, and with a whole host of other factors, for example, giving methicillin to farm animals, the bacteria were able to adapt, and they were able to resist methicillin, and that's where we get MRSA from, methicillin-resistant Staph aureus. So how do they become resistant to the methicillin? They produce different PBPs. So here's our penicillin binding protein. And let's say our penicillin binding protein has undergone an alteration. So its shape has changed a little bit. That shape change allows it to interact with the substrate molecule, but that change is enough so that it can't interact with the drug. That's how changing the penicillin binding protein can lead to resistance. On this slide here, one source of MRSA was from pigs. Humans gave Staph aureus to pigs. We then grew pigs in these very packed environments, unhealthy environments. We gave them antibiotics in their food, a lot of drugs, a lot of bacteria, tight living conditions where the spread of bacteria is all but assured. That leads to selection of those bacteria that can survive. And not too shortly after that, we have resistant bacteria. On to slide 27, cephalosporins. So you can see here on the slide there, you can identify the square shape in our beta-lactam drug, the beta-lactam ring. Cephalosporins are beta-lactam antibiotics that have the ability to resist some beta-lactamases. There are different generations of drugs. They've been chemically altered, first generation and second and third and fourth. Typically those drugs begin with names like Ceph, CEPH or CEF. Currently there is a fifth generation cephalosporin that is used against MRSA. Some of these drugs can be given synergistically with a beta-lactamase inhibitor. So, what's a beta-lactamase inhibitor? It's exactly what it sounds like. It is a drug. So, we have our penicillin binding protein down here. We have our beta-lactam drug over here. We want our beta-lactam drug to get and stick into our penicillin binding protein. But the bacteria has a beta-lactamase. It's going to come in and snip up the drug. If we can then send it in a second drug that interferes with the action of the beta-lactamase, then the beta-lactamase can't snip up our drug, and our drug can stick to the penicillin binding protein, and it can interfere then with the production of cell wall production. Our next type of beta-lactam antibiotic on slide 28 are carbapenems. These are, again, if you look at the structure there, you can see the beta-lactam ring. These are drugs that are not inactivated by a whole number of beta-lactamases, in particular those extended spectrum beta-lactamases. Our carbapenems are a type of drug that are typically reserved for those drug-resistant strains. So you try out the first drug and you find out that the bacteria are resistant. And then maybe you'll have another drug, and then sooner or later, if you keep hitting resistance, you might switch to a carbapenem. And then there's a chance that the carbapenem will work. Now we want to keep this kind of drug back We don't want it out there treating everybody, because the more a drug like carbapenem is out there, the more the bacteria are exposed to it, and the easier then it is for those bacteria to become resistant. And indeed, bacteria have become resistant to carbapenems, CREs, or carbapenem-resistant Enterobacteriaceae. And once a bacteria becomes resistant to carbapenems, then very likely it is resistant to all other known antibiotics. On to slide 29. We're still looking at drugs that inhibit cell wall synthesis, but we are moving away from the beta-lactam antibiotics. We're now looking at other drugs that affect cell walls. And our first kind of drug on page 507 are glycopeptide antibiotics. So these do not have that beta-lactam ring, and they don't work by interfering with penicillin binding proteins. Probably a drug you may have heard of is vancomycin. Vancomycin is a glycopeptide antibiotic. What we have here is on the slide there, we have a view of what the drug is doing. So it blocks peptidoglycan synthesis. We have our side chains. We have our molecules sticking to the side chains, and as they stick to those side chains, the enzyme, the bacterial enzyme that's supposed to come in and fix those chains together is blocked. You can see those little blue kind of clamps. Now you can also see that it's able to become resistant when the clamps can no longer clamp on to the onto those side chains. So vancomycin blocks peptidoglycan synthesis, but it doesn't block it in the same way as our beta-lactam antibiotics. So this is where we look at that idea of getting drugs to different places. This is a drug that is usually administered via IV because it isn't uptaken through the intestinal tract very readily. So even though it can survive the journey through the acid of the stomach, once it gets into the intestine and is broken down, it doesn't get absorbed. So much better to give it as through an injection. Important for treating gram-positives that are resistant to other beta-lactam antibiotics, and good for treating severe C. diff cases. Another antibiotic that is of last resort. Bacteria are becoming resistant to most of our drugs. Vancomycin is one of those drugs that we try to hold back and only use it when we've had no other options, so that we can limit its exposure and limit the ability of microbes to become resistant. We are now getting vancomycin resistant Staph aureus, so no longer MRSA, VRSA. Last for this section has been pretty long, so bear with me. Another drug that inhibits cell wall synthesis, again does it in another different way is the bacitracin. And you'll see in the picture on the slide there, all of the components of our bacterial cell walls, the sugars and the amino acids, also all the stuff that builds those components into the cell wall, they're all generated inside the cell. So we have our DNA and our genes. And then our genes are turned into proteins. And our proteins do some kind of chemical work and they build a subunit. Maybe they build a NAM subunit. But our wall is being built outside of the cell. So we have to get this subunit across the cytoplasmic membrane and kind of get it to where it's needed. So our bacitracin is a drug that blocks the ability of the bacteria to export these subunits. Now it turns out the bacitracin is quite toxic, most likely because the mechanism that a bacteria has to move things across its membrane is probably very similar to the mechanism that we have to move important things across our membranes. So if you block the ability of the bacteria, you're probably going to block the ability of the eukaryotic human cells. So bacitracin is typically available as a topical application, particularly as applied to skin. And you may find it in many over-the-counter kind of antibacterial skin ointments. Okay, so a lot of stuff in that section there. Beta-lactam antibiotics work by gumming up penicillin binding proteins. They only work against actively growing cells because we are interfering with the building process. Once the wall is built, we aren't going back and damaging it. Gram-positives and gram-negatives, different in their structure. Gram-positives have the peptidoglycan on the outside. All of the stuff that's building that peptidoglycan is on the outside, and all of that stuff is readily available to our beta-lactam drugs to interfere. Our gram-negatives have that outer membrane as an additional barrier. Beta-lactamases, these are enzymes that the bacteria makes that hope to snip up the beta-lactam ring of those beta-lactam drugs before they can interfere with those penicillin binding proteins. MRSA, methicillin-resistant Staph aureus. Methicillin is a kind of penicillin. Methicillin is a penicillinase resistant, but through various uses, bacteria have been able to change the shape of their penicillin binding proteins so that the methicillin no longer sticks. And that's given us, particularly in S. aureus, we now have methicillin-resistant S. aureus. Carbapenems, a drug that is held back, is used as kind of a last resort for someone who isn't responding to other medications. Again, been held back because we don't want the microbes exposed to it. The more we expose microbes to these drugs, the easier it is for them to become resistant. And carbapenem-resistant Enterobacteriaceae is a set of bacteria that are resistant to these kind of last line of defense drugs. Vancomycin, not a beta-lactam. It works by interfering with the ability of other enzymes to put the the cell wall together. And bacitracin, a different kind of drug once again. It interferes with the ability of the bacteria to export pieces of the wall across their membranes. Okay, that should be enough for this section.